So our next speaker is Dr. Tim Hunt, Nobel laureate in physiology or medicine, emeritus group leader of the Francis Crick Institute. While for most, science is often considered a knowledge-based business, but to Dr. Hunt, science is all about the world's many mysteries. For it is the mysterious side of things and the quest for understanding of the unknown that leads people to wonder and explore. From his own scientific experiences, Dr. Hunt came to realize that oftentimes seemingly simple questions do not necessarily have simple answers. Yet progress in science depends on questioning, on curious people doing experiments to find things out and get answers as to what yet is not known. We now look forward to hearing from Dr. Hunt on pain and suffering, the agony of not knowing.
Huh. Well, well, well. So, thank you, Laurie, and thank you, Bent, for inviting me. And thanks very much to the young man up there who is the best audio-visual helper I've ever encountered anywhere in the world. This is such a portentous kind of symposium that I really wasn't sure why they invited me, and B, what I should say. But I think I should start by explaining sort of about Nobel Prizes and what they're all about, I suppose. So my wife says she's not very respectful. If I can win a Nobel Prize, anybody can. And it's kind of true, actually, because, I suppose I always wanted to win a Nobel Prize, and I think it's sort of a good thing to want to win, because why would you not want to find out the most important truth that you're the only person in the world to figure out, that everybody wants to know the answer to? I mean, this seems to me a very noble aim, but it's easier said than done. And I will explain where this pain and suffering thing comes from a little later on. So people think that Nobel Prize winners are all geniuses. We are not. I'm especially idiotic when it comes to physics, as I will now explain. So here is Schrödinger's kitten. This is my daughter, Celia, aged about 7 or 8, I think, and the cat here, Minky, was eaten by a fox a week after this picture was taken. Now, so getting that phone call from Sweden is really sort of very, very peculiar. Because, before you were just a scientist sort of struggling with your group and things that weren't working properly and stuff, and then somebody calls you up and says you're a Nobel laureate. Genius, walk on water, not a problem. 2 places at once, easy. But Celia asked me this question at bedtime. Daddy, it was about 2 weeks after I could walk on water and be in 2 places at once. Daddy, why is the ceiling opaque? So I looked at the ceiling, I thought I knew the answer to that, but then I looked out of a window, and I thought, oh my God, how the hell does light get through glass? Now, for some reason, I'd just been reading Newton's work about light, and Newton says that light is made of little particles. So was the stuff that windows are made of, was there enough room for these little things to get through the cracks in between? That somehow didn't seem very likely. And then I thought, well, glass is a frozen liquid, so maybe it's to do with liquids. And light does go through air, and it goes through water, but oh, wait a moment, mercury is a liquid and it doesn't go through that. You make mirrors out of mercury, so that's not the answer. I thought about carbon, and carbon can be black, or it can be gray, but it can also be totally white. So then I had a look at, I read Einstein's famous 1905 paper. I can't read German, so I got it translated. And I thought this was absolutely amazing. Basically, you know, it is light. You sort of know, is it little particles or is it little waves? A couple of days ago, I went to, I looked at Richard Feynman's lectures online. I would strongly recommend them, actually. And in the first one, he says, light is waves, and he compares it to people jumping into a swimming pool. And in the second one, he says, no, light is particles. So that wasn't terribly helpful, but I found this very helpful, because if light was waves, how could we see the stars? Because by the time the waves reached us, they'd be so faint that we wouldn't be able to see them at all. So, you know, this according to the assumption here, light rays spreading from a point, the energy isn't distributed continuously over ever-increasing space, because a finite number of energy quanta that are localized in points in space, move without dividing, and can either be absorbed only as a whole. So, you know, this doesn't help you, however, understand how things get through glass. And then I had to give a talk at my old school to open a lab, and on the bench was a magnet, a coil, and a galvanometer. And so I, you know, being of an expert, I pushed the magnet into the coil, and sure enough, the galvanometer moved. And then I thought, well, I wonder if I turn the magnet around, push it in, will the galvanometer's needle work in the opposite direction? And I was absolutely thrilled when it did. And you sort of knew about this. I mean, I'd been doing this kind of stuff since I was a little boy. But I hadn't really understood properly that moving magnets cause currents to flow, and that flowing currents cause magnetism. And this, you know, what's it got to do with light? Well, everything, as it turns out. I mean, this is an extraordinary thing. This is the great Michael Faraday, who has an amazingly intuitive understanding of all this stuff. You know, although we know nothing of what an atom is, yet we can't resist forming some sense of a small particle which represents it in the mind. And though we're in equal, if not greater, ignorance of electricity, so as to be unable to say whether it's a particular matter or matters or mere motion of ordinary matter or some third kind of power or agent, yet there is an immensity of facts which justify us believing that the atoms of matter are in some way endowed or associated with electrical properties to which they owe their most striking qualities, and amongst them their mutual chemical affinity. Fantastic, absolutely fantastic. But it was till Maxwell came along, who was a genius mathematician. Actually, he was the second best mathematician of his year in Cambridge, who figured out the theory of electromagnetism and worked out that actually light was an electromagnetic wave. Now, I realized when I looked at Maxwell's equations why we'd never studied how light got through glass in school, because it's very, very complicated. Maxwell's equations, you don't get to them until you're in about the third year of physics in Cambridge, because they're very, very difficult to understand, and certainly not the kind of thing that the average school teacher or the average school child could understand. And then but, you know, he figured it out. It seems to have strong reason to conclude that light is an electromagnetic disturbance in the form of waves. I mean, this was an absolutely fantastic, towering achievement of the 19th century. And conducting media are shown to absorb such radiations rapidly and therefore to be generally opaque. So that, in a way, is the answer to Celia's question. Well, I still didn't really understand, though, how light got through glass. And I asked, it was funnily enough at the British laureates lunch for the laureates in Stockholm in 2001. I was sitting next to Aaron Klug, and I said, Aaron, how does light get through glass? He said, well, Tim, you really need to understand Schrödinger's equation. At which point I knew I was lost. So here we are. Here's Schrödinger's equation. Looks very simple. Physicists love simple equations because they explain things completely. And this is Schrödinger's tomb, actually, in Alpbach in Austria. And it's really quite amusing. I don't know if you can see, but whoops, daisy. The original sculptor got it wrong. There's a dot over that second psi. So what does it mean? Well, I, of course, as you know, is the square root of -1. Not a promising start. The only exam I ever failed in my life was an advanced maths exam, which had complex numbers and vectors and things in it. And, so h bar, obviously, Planck's constant divided by 2 pi. Psi dot, the rate of change of the wave function with respect to time. How many of you actually out there know what a wave function is? I bet some of you do. Yes, look, hands go up. Great. You see, I don't, which means that I really don't understand. Equals h, the Hamiltonian. Hamilton was a very famous Irish mathematician of the mid-19th century, times the wave function. Well, you know, what has that got to do with the price of onions? Didn't really help at all. And then we found Dirac, who worked out the relativistic form of the Schrödinger equation. The problem of getting the interpretation proved to be rather more difficult than just working out the equations. So, you know, things are getting pretty serious. And then we have Feynman. Such an accurate theory of the interactions of photons with electrons that if you were to measure the distance from New York to Los Angeles, it would be specified to the width of a human hair. I mean, that is one super, super accurate theory. So all of this is really by way of saying there are some people who kind of understand, who can describe how light gets through glass, but for the most of us, it's a perfectly reasonable question, but it doesn't have a simple answer. I mean, it led me into all these understandings of, I began to really sort of get to grips with the complexities of quantum mechanics. Anyway, the real point of this talk is about this. The business of science is about finding things out. And as I say, it's easier said than done. And the other thing, we're supposed to be worrying about the future, and the trouble is that we don't know what the future is going to bring. In forecasting the future of scientific research, there is one quite general law to be noted. The unexpected always happens. So one can be quite sure that the future will make any detailed prediction look rather silly. And here is my friend Adam Smith interviewing Honjo for on the board of his Nobel Prize. So you never really know which way things will go in research. You didn't set out to discover a cure of cancer. No. Well, so biology is such a complicated system, it's totally different from engineering. We can't design. Most people tried to find a therapy for cancer, but all failed. And I myself, I never expected my research would lead to a cancer therapy, but in a sense, I'm very fortunate that I thought about it. You know, you have to try many things, and if you're lucky, you can hit, but you have to pursue. That's my feeling. I think that's very nice. So people ask me for advice, you know, what should I do? And I tell them that you have to keep your eyes on the horizon, but your feet on the ground and your nose to the grindstone. But the most difficult thing, I think, in my experience, is finding a good problem to work on. And this man, Peter Medawar, wrote essays that inspired me very much when in my younger days. And he wrote an excoriating review of The Art of Creation by Arthur Koestler. If politics is the art of the possible, research is surely the art of the soluble. In fact, I think the collection of essays was called The Art of the Soluble. No scientist is admired for failing in the attempt to solve problems that lie beyond his competence. And here's Steve Weinberg. In the real world, it's very hard to know which problems are important, and you never know whether at a given moment in history a problem is soluble. And here's somebody else I heard give a talk, which I thought was absolutely marvelous. Number theory, this is Robert Aumann, an economist. Number theory fascinated me because the problems are very natural. They're simple to formulate. A school child can understand them. But the solutions are very difficult and deep. They require years of university study even to begin to fathom. And the whole subject is absolutely useless. I especially like that. But he goes on to say science is exploration. Exploration for the sake of exploration and for nothing else. We must go where our curiosity leads us. We must go where we want to go, and eventually it's sure to lead us on to the beautiful, the important, and the useful. I think that's a beautiful expression of what it's about. And here's my old nemesis, Sidney Brenner. What we're looking for is innovation, something new, and you won't get something new if you work within a completely established mode of work. We need people to stray off the standard path and ask new questions. And the important thing about research is that it's about going where none have gone before, so you have no other recourse but to teach yourself. That's why research is so difficult. You don't know which way to turn, and you're on your own, out there, asking questions of nature, and you don't know if there's an answer. You have a feeling that there probably is, but you can't be sure. So it's always a difficult and uncertain business. And I've noticed that scientists' mood varies very much according to whether things are going well or whether they're going badly. And here's a great director of science, Max Perutz, who was in charge of the famous MRC Laboratory of Molecular Biology we'll get to in a second. Creativity in science, as in the arts, can't be organized. It arises spontaneously from individual talent. Well-run laboratories can foster it, but hierarchical organization, inflexible bureaucratic rules, and mountains of futile paperwork can kill it. Discoveries can't be planned. They pop up like puck in unexpected corners. So here is Max's old lab. The new one is much fancier and bigger, not far away from the old one. And I had the good fortune to, at one stage in my life, I burned the lab down and we had to move. And we occupied a sort of unused hematology teaching lab, which was just opposite. And Max very kindly said we could use his tea room, which was the most amazing gift that you could give to a young scientist, because the tea room was full of my heroes, Fred Sanger, Francis Crick. Jim wasn't around much at the time, but I met him later, Max Perutz, Sidney Brenner, and a string of Nobel laureates, including people from my own generation, finally. And, you know, it's sort of interesting because it wasn't, you know, we didn't think of ourselves as genius scientists. We had role models, for sure, who were, but one of the things about the role models, how very different they all were. I mean, Fred, who won 2 Nobel Prizes, was a very sort of quiet, modest man. You might have taken him for the janitor or the gardener or something like that if you met him. Whereas Francis was very ebullient and full of always enthusiastically explaining things. Although he taught me one very valuable lesson. He went to a lot of seminars because he didn't do experiments himself. He liked to hear what people were saying, and he often asked questions at the end of the seminar. And he often asked what to me were quite ignorant questions, and it became clear to me that the reason why he asked questions because he was uncertain, he didn't know himself, and he wanted to be sure he got things straight. He wasn't trying to show how clever he was or to show up the speaker. He just wanted to make things clear, and that was very good. So, you know, they were so different from one another. I mean, Sidney was absolutely terrifying because he couldn't resist a joke, and you know, if it was a joke at the victim's expense, it didn't matter, and so he could be very cruel. But a wonderful scientist and a wonderful man. I sort of really came to appreciate that very much later in life. His heart was 100% in the right place, and he came from extremely humble origins. His father was an illiterate maker of shoes in South Africa. And you have to grab your opportunity. This is my other daughter, Agnes, who liked to skate. And when at school, when it froze, the whole school stopped and we just went skating because it wouldn't happen again. I think it's very important to sort of grab your opportunities. But after Francis Crick and Sidney Brenner had worked out how the idea of messenger RNA and DNA makes RNA makes protein, and the genetic code was solved, they had a problem, which was what to work on next. So Francis went to work on consciousness, and I still don't really know whether he made any serious contribution to the understanding of consciousness. It's not even clear that consciousness is a scientific problem. It might be more of a philosophical one. And Sidney wanted to work out how brains work, so he took the simplest brain, the brain of a nematode worm with 302 neurons, and he thought, if I get the wiring diagram, then I'll understand how it works. So he worked out the wiring diagram of the worm, and people still don't know how the worm wiggles. I mean, I think there's a really serious problem with all this. I mean, it's very good to see the connectivity in the brain and know what all the different parts do, but it's a very far cry from working out how it works. And if I were a neurobiologist, I think I would try to work on something really simple like a worm. However, Sidney, fortunately, was awarded, he should really have been awarded 2 Nobel Prizes, one for the discovery of messenger RNA and the second one for his work on the worm. But the Nobel people were very clever because Sidney's students worked out how programmed cell death worked, and so it was a sort of apoptosis prize, really, in the end. So how do you get here? How do you get to be a wonderful scientist? Well, it boils down, I suppose, to education. And here is, as far as I know, the only essay on science that George Orwell ever wrote. And I was very taken with this. I think it's absolutely right. Scientific education ought to mean the imparting of a rational, skeptical, experimental habit of mind. It ought to mean acquiring a method that can be used on any problem one meets, and not simply piling up a lot of facts. The idea that science means a way of knowing the world, looking at the world, and not simply a body of knowledge, is in practice strongly resisted. How true that is. I mean, those 2 daughters I showed you, they're neither of them scientists, and one of the reasons I think they're not scientists is because they were taught science in a horrible way by people who didn't really understand what they were talking about. And I remember at one time trying to help Agnes with her homework, and it was about the adaptation of pine forests to the environment. And, it was all about sort of learning the shape of the pine tree. I tried to sort of explain it. The book didn't really explain properly how the pine trees were adapted to living in the north of the world. And, oh, Dad, shut up. Don't just tell me what the answer is. Won't do. And at one time, this is amazing, I was asked to comment on a syllabus, and I went to the Ministry of Education, and there were all these science education experts, and I'd read not only did I read the biology one, which I didn't like very much, but I read the physics and chemistry ones, and I was a little alarmed because there was no mention of atoms in the chemistry thing. So I poked up my hand and said, why is nothing about atoms in the chemistry syllabus? That's physics. What? Unbelievable. Unbelievable. And then this is one of the practical things. Look at this. What? How could you use a water bath unsafely, I wonder? I suppose throw it. Oh dear, oh dear, oh dear, oh dear, oh dear. And see, I had a very different kind of teacher. This is my first science teacher, and he was terrific. He understood the mind of the 12-year-old boy perfectly, and his instructions from the headmaster, just get them interested. Isn't that fantastic? So, we took radios apart. I mean, in those days, of course, you could understand a what a radio. Nowadays, you know, even if I were to open this thing up and looked inside, you wouldn't have a clue what was going on. So that's bad luck for you guys out there. When I was a boy, you know, there were parts to radios and TVs and things, and you could sort of see what was going on. Anyway, but of course there is a question of whether you really need to teach science in schools at all. And I once sat next to Tony Leggett at a dinner in Singapore. And this is remarkable. I mean, so this guy never did, he had no science in school whatsoever. In fact, he had no science in school, he had no science in university. He's a Nobel Prize winner for the studying of helium-3 close to absolute zero, sort of quantum thing. So how did he do it? Well, I did indeed consider the possibility of going into pure mathematics, but rejected it on the grounds that in mathematics, almost by definition, to be wrong means you're stupid. I wanted the possibility of being wrong without being stupid, of being wrong for interesting and non-trivial reasons. Physics seemed to me to fit that bill. And while I had no training in the subject, what happened was he decided to, he thought of doing philosophy as well, because he read Greats in Oxford, which Celia, the first girl, actually did. She's ended up designing websites because it turns out that Greats, which is the study of Latin and Greek philosophy, language and stuff, which used to get you could be a spy with that. Nowadays you have to be a genius mathematician and computer expert to be a spy. But anyway, it served Leggett very well because he explained to his tutor in physics that was what he wanted to do, and the physics teacher, Don, gave him a book of physics and said, why don't you work on these problems? So he worked on the problems. He did very well at them because he was quite good at maths. And you know, and the only problem was he didn't have a grant because he'd already had his 4 years at Oxford. And then he was saved by the Russians who launched Sputnik. And so suddenly Britain wanted more physicists, so he got his 1-year grant to study physics, and you know, and then the rest is history. So maybe it would be better if people didn't really learn any science in school, you know, because then that would save them from how to use the water bath safely. And in terms of sort of humanity, this is Bruce Alberts, who I got to know quite well because he was the author of a textbook I helped making the problems for. And he was editor of Science magazine and for a long time president of the US National Academy. I came to realize that science is much more important for the world than most scientists suspect. The scientific values of honesty, respect for evidence, openness and tolerance are critical for every nation. I think that puts it very well. So just a word or two about what I did. Normally I talk about this in much more detail. So here this now explains the title of the talk, Pain and Suffering. This guy here is Stanley Cohen. This one here, this is me in 1977. And Stanley had discovered epidermal growth factor. And he then studied the epidermal growth factor receptors. That's what he was doing that summer, actually. And he was kind of stuck, actually. But then later discovered that the epidermal growth factor receptor was a tyrosine protein kinase and won the Nobel Prize, I forget which year, I think in the early 80s, about a decade later than this picture was taken. And I went to see him. Our paths didn't cross again after that summer. They were great friends. We used to play tennis together. We were very evenly matched because Stanley was a polio victim and couldn't... couldn't run very fast. Now he's just terrible. We used to play after dinner before the sun set. It was great. So I went to see him in his lab, and I was very surprised. He was by this time sort of 75 years old or something in Vanderbilt University. And I was surprised to find him in a white lab coat in the cold room. And I said to him, Stanley, what are you doing? You know, you don't need this. He said, Tim, he said, it's pain and suffering. Pain and suffering. I can't stand not knowing what the answer is. And I pushki around, he said, and every 10 years ago or so, I find out something that makes it all worthwhile. And I thought that puts it beautifully. You know, it really hurts not knowing. So why was I in Woods Hole in the summer of 1977? Well, I had solved my problem, which was how the synthesis of heme and globin were coordinated. And I was looking around for a new problem, and I remembered a talk I'd heard many, many years before about sea urchin eggs. And here I am getting sea urchin eggs out of a female sea urchin called Arbacia punctulata. And you give them a 12-volt alternating current electric shock, and they shed their eggs. And I was trained as a biochemist, and sure enough, I confirmed the result, which had been known for many years, that when you fertilize the eggs, there was a big increase in the rate of protein synthesis. Time along the bottom, protein synthesis measured by radioactive incorporation of labeled amino acids into proteins up the side. And you can see there's sort of a 10 to 20-fold increase in the rate. Unfortunately, it looks dramatic, but what that is from almost no ribosomes being active to about 0.5% of the ribosomes being active. And it wasn't at all, we never did solve the problems. But along the way, I started wondering why protein synthesis was necessary. Why did you have to turn on protein synthesis? Well, you might think, you see, these eggs aren't actually going to grow for the first bit. All they do is divide. If we look at this, we should be able to see them. If I add some sperm, they divide amazingly rapidly. The first division after about an hour, and then each subsequent division about every 15 minutes. And notice, incredibly synchronously. You don't have to do anything except make sure the sperm is properly mixed up with the eggs to things. So, you know, what we did the first 2 or 3 summers I was there was just to learn how to deal with these eggs, how to get the concentration of sperm right, how to... I lost my sense of smell in the process because it turned out that the sperm has huge concentrations of proteases in the acrosome. I'm not quite sure why. Maybe it has to use those to burrow through the surface of the egg. But anyway, so we got a phenomenon known as upper gel wipeout. All the high molecular weight proteins were dissolved. The proteases were more resistant to SDS than the... Anyway, so we learned how to run beautiful gels of fertilized sea urchin and later of clam eggs also. And then I did this amazingly simple experiment. You see, here we just look at the overall proteins, but what happens if you look to see what proteins are actually made? And nobody, I mean, it was extraordinary. Anyone could have done this experiment in the 10 years previously where the methods had all been worked out, but it was such a dopey, simple experiment that nobody actually had. And I think you don't need to be a Nobel Prize laureate to realize that something very interesting is going on, because you can see that these are clam embryos, actually. There are 2 proteins here which get made very strongly early on and then disappear, and then they come back again, and then they disappear. And I was just lucky because I was the first person to see that, and I was the first person to see that because I knew how to analyze these proteins on gels. And I was in the habit of taking timed samples. And if you see, you need to watch something go away, you obviously need at least 2 time points before and after it's gone away. So I saw it, and I immediately knew that I'd stumbled on something very, very important. And also something, as I'll say in a minute, impossible. So after a long time, it took ages to clone and sequence and figure out what these things were. It took about 4 years. But it turned out that That's my protein on the right there, cyclin I called it, because I was a very keen cyclist at the time, and the thing went up and down. You know, it was sort of one of the things you can call your protein, if you're the discoverer of a protein, you can call it anything you like. There's no international commission on protein names. It's great. And it turned out that it was really simple once we understood that cyclin accumulates, it joins on to CDC2, that's the protein on the left, which is a protein kinase, and everything, and that was the enzyme that catalyzed cell division. I mean, that was another thing that Stanley taught me, actually. I mean, the idea that you could have an enzyme that catalyzed a process as complicated as cell division was quite extraordinary. I mean, enzymes usually do very boring things, like convert glucose to glucose 6-phosphate. It doesn't. The idea you could have an enzyme that catalyzed cell division was a revelation to me. And I think this is a very good example of something that another great hero of mine, Theodore Bovary, said that the disclosure of a single new fact suddenly can illuminate a quite distant area, hitherto dark. Just one little tiny clue gives the whole game away. And that was the thing, you see. Cell division needs new protein synthesis. That was known. But the idea that it also required selective and highly regulated protein degradation had never been proposed, had never been even remotely contemplated, because at the time it would have been considered to be impossible. And biologists don't like to think about things that are impossible. I mean, actually, if you want to win a Nobel Prize in biology, the best thing is to do something that people think is impossible. For example, my teachers told us that it would be impossible to sequence DNA. Huh? Theoretically impossible, because the way you sequence things was the way that Fred Sanger sequenced proteins, and that wasn't appropriate for DNA, right? But they reckoned without Fred Sanger, it wasn't impossible, it could be done. And now people increasingly find even better and cleverer ways of sequencing DNA. So there was a Nobel Prize for sequencing DNA. There was a Nobel Prize for discovering the structure of the ribosomes. Another thing that I was taught was theoretically impossible, because A, ribosomes would never crystallize because they were too heterogeneous and too big and da-da-da. And even if you solved them, you wouldn't be able to solve the structure because they were too complicated. Well, that was well and good, but actually computers and synchrotron illumination got better and better and better, so it was no longer impossible. But I think that's another good example. And indeed, Jack Szostak and Liz Blackburn's prize for artificial chromosomes, or telomeres, actually, I think it was, is another good example. And whoever dreamt that you could make a synthetic chromosome? I mean, this was terrific stuff. But I was just lucky. I mean, I just sort of stumbled on the truth. But I think the reason why it was a Nobel Prize was because beforehand it was nobody even, never crossed anybody's mind. And it made everything much simpler and obvious. And here we actually have a picture of Feynman, and I like this very much. I think it's so true. When you get it right, it's obvious that it's right, at least if you have any experience, because usually what happens is that what comes out, more comes out than goes in. Your guess, in fact, is something is very simple. If you can't immediately see that it's wrong and it's simpler than it was before, then it's right. The inexperienced and crackpots and people like that make guesses that are simple, but you can immediately see they're wrong, so that doesn't count. Others, the inexperienced students, make guesses that are very complicated, and it sort of looks as though it's all right, but I know it isn't true, because the truth always turns out to be simpler than you thought. But boy, is it unsimple when you don't know what it is. And finally, there is only one way to find things out. And, just do the fucking experiment. Thank you very much.